This method allows to elucidate the microscale ecology and behavior of microorganisms navigating dynamic chemical gradients, and to uncover their hidden trade-offs, nutrient kinetics, and population dynamics in ecologically relevant microenvironments. By combining microfluidic technology with photolysis, we generate controlled chemical pulses where localized chemoattractant becomes suddenly available at the microscale to measure the chemotatic response of microbial populations first exposed to unsteady chemical gradients.
Design the channel using CAD software and print it onto a transparency film to create the photomask. Fabricate the master by soft lithography in a clean room according to the manuscript. Prepare a PDMS mixture by combining the elastomer with its curing agent at a 10:1 ratio in a 40 milliliter beaker. With a plastic knife, mix vigorously until the liquid is homogeneous. Immediately degas the PDMS mixture in a vacuum chamber for 45 minutes at room temperature. To expedite the process, periodically release the vacuum in order to burst the bubbles that form at the interface. Remove dust from the surface of the master with a pressurized cleaner. Then pour the degassed PDMS mixture onto the master and bake it in an oven at 80 degrees Celsius for 2 hours. Cut the PDMS with a blade around the microstructures at a distance of approximately 5 millimeters, and then carefully peel the PDMS from the master. Punch holes to serve as the inlet and outlet of the microchannel. Seal the microchannel with adhesive tape to prevent accumulation of dust.